So as Ashley mentioned, I am the co-leader of the Neuropathology Core and Brain Bank in the Duke-UNC ADRC. So our involvement in AD research is mostly during brain donation autopsies. We go into the room to dissect brain tissues at the time of autopsies. I provide the neuropathological diagnosis for clinicians and the family. My lab manager and technician goes in to collect CSF and brain tissue and for long-term storage, and we maintain a database with all the clinical and neuropathology information that we can provide for investigators. And another important function, of course, is we are a biorepository. And so we receive requests for tissue and data all the time, and we will evaluate and complete these tissue requests. And we also provide consultation for study design and for neuropathology. And we also are a histology core, so we can cut sections for you, do most of the standard stainings. So a successful brain donation program requires a lot of people, and neuropathologists and the brain bank are really at the very end of this process. So I won't go through the details about how the patients are recruited and how important it is to have like a good relationship with the family and for longitudinal follow-up. But just at the time of the patient's death, for an autopsy to happen smoothly requires a lot of coordination. And it is really thanks to the clinical coordinators. They are the ones usually, they serve as the point contact for the family. And then they have to contact all these different people, including the sedum care at the hospital, the autopsy team. They have to arrange for body transport. They have to contact the brain bank, and they need to make sure of the funeral where the family wishes to send their body to the funeral home. So it really, for an autopsy to occur, it takes almost 2 hours to coordinate an autopsy. And they have to be well-trained and be able to do it in a very sensitive and understanding manner. And we are the ones who go in at the time of autopsy to take our fixed tissue for neuropathology diagnosis, and then we go in to bank the CSF and frozen tissue for our biorepository. So at the time of autopsy, we take out the brains, and the brains are separated in two halves. So one half will be fresh, and my lab manager, he will go in and cut the brains fresh, which is pretty difficult because a fixed brain is really mushy and very soft. So he has to be very skillful and very careful about handling fresh tissue. And then the other half will be put in formalin for 2 weeks to be fully fixed for neuropathology workup. So my role really as a neuropathologist is really focused on diagnosing neurodegenerative diseases at autopsy. So I will give you some introduction of what I do, so that you could probably understand a little bit more about these neuropathology reports. So the first thing starts with gross examination, and there are a lot of things to look for. One important aspect is to look for the pattern of atrophy. So for instance, in many cases, there will be a global atrophy, and which is typical for AD, although in most cases, it starts from the frontal and temporal lobes. In some cases, there will be a very accentuated atrophy focused on the frontal lobe and temporal lobes, and these are characteristic of the frontal temporal lobar degenerations. Some diseases are characterized by a profound asymmetry. In this case, you can see one hemisphere is a lot more atrophic than the right side is a lot more atrophic than the left side. So this is characteristic for cortical basal degeneration. So just by gross examination will give you an idea of what we're likely to deal with. Also, um, besides the pattern of atrophy, we also look at the color and the distinction between these different structures. So for instance, in this particular brain, this is the midbrain, and we're looking at the substantia nigra here. You can see that in a normal control, it will be well pigmented. There will be, it will appear as a well-defined black line. Whereas here, the black pigment has disappeared and it just has this kind of yellow haze. Also notice that there is significant atrophy of the midbrain compared to the pons. And then there's also discoloration and atrophy of the subthalamic nucleus, lack of clear distinction between the putamen globus pallidus, shrinkage of the dentate nucleus relative to the other parts of the brain. So on gross examination, this is characteristic for progressive supranuclear palsy. In some cases, we also get not only dementia patients, but also some movement disorders. So this is another kind of characteristic brain that we could sometimes see. So again, you see the loss of pigmentation in the substantia nigra. And here you have this gray, bluish gray discoloration of the putamen, which is very characteristic for this entity. So this is a case of multiple systems atrophy. So by carefully looking at the gross, we could already get some idea of what entity we may be dealing with. The next thing is that we cut the brains sequentially, and we usually cut the cerebral hemispheres coronally. Brainstem and cerebellum we cut in a different plane. Then we go into these different structures and sampled each region. So for instance, this is a cross-section through the thalamus. Okay, so this is thalamus. And then here we have this almond-shaped structure that is the subthalamic nuclei. There is some pigment here. So this is the substantia nigra, although it is in the midbrain, but it also extends up into a little bit into the thalamus or subthalamic region. Here I'm showing another coronal section, and this is more posterior. And so this is in the occipital lobe. On the medial side of the occipital lobe, there is this structure called the calcarine fissure. And along the calcarine fissure sits the visual cortex. So these are all important brain structures that you want to sample. So I would go in and cut these sections, put them into these plastic cassettes, and hand it over to the histology technicians. And then they will cut like 5-micron sections on the microtome and then do the staining. So for instance, so for this one, this section is from here. This is the visual cortex. So this is very characteristic of the calcarine fissure. This is what it looks like on a microscope, on very low power microscope. This is the hippocampus. You see the curve here, the dentate nucleus. Oh, sorry, dentate nucleus goes in this direction. Then you have the hippocampus proper going into the entorhinal cortex, and then this is the adjacent temporal cortex. So this is pretty much mirroring what's shown here. This is the gross image. This is what what it looks like after they cut 5-micron sections and stain it with Luxofast blue. And here is very anterior basal ganglion. You have the caudate nucleus and putamen, and this is internal capsule. So this is again mirroring what you see here. This is putamen, internal capsule. Oh, sorry, caudate, internal capsule, putamen. So we rely on these set of sections to do our neuropathology workup. And I routinely section these 12 brain regions, and it is based on our understanding of how these different neurodegenerative pathologies spread. So for instance, for amyloid plaques, we know that they start in the cerebral cortex and then spread into the limbic system, including the amygdala, hippocampus. And then lastly, in the later stages, they go into the brainstem and cerebellum. The neurofibrillary tangles, they have a different pattern of spreading. So they would start in the entorhinal cortex and the locus ceruleus, which is in the brainstem here. Then they would spread into the hippocampus and adjacent temporal cortex, and then slowly go into the frontal cortex, and lastly into the occipital cortex. Lewy bodies, they start in the brainstem. They also go into the limbic system, and then lastly spread into neocortex. So we basically have to sample all these brain regions and do appropriate staining to look for these different protein aggregating inclusions. And and based on what our understanding of how these pathologies developed, finally give them a pathologic stage. So which I will talk about a little later. So for instance, for Alzheimer's disease, we all know that the pathologic hallmarks are the amyloid plaques and the neurofibrillary tangles. So there's many different ways of looking at amyloid plaques. One way is to do immunostaining for amyloid beta, or APP. And here, whatever stains for amyloid beta will show up as brown, and then the blue is just a counter stain as a background contrast, so you can see the structures more clearly. So the amyloid beta, they will label all kinds of different plaques, and for neuropathologists, we like to characterize them as, for instance, these are called the dense core plaques, where you have a very dense core in the middle, a rim of empty staining, and then more hazy brown staining outside. There are also other type of plaques, like these are called the diffuse plaques. They don't have this dense core in the middle. Then you can also have other type of diffuse plaques, and also you can have amyloid deposition in the artery. So these are called cerebral amyloid angiopathy. They can cause damage to the blood vessels and cause predeposition for bleeding. So the staging of amyloid beta basically follows this idea that amyloid beta starts from the cortex, goes into the limbic system, and then lastly into brainstem and cerebellum. So this is described by Dietmar et al. Back in the 90s. So we, for the amyloid beta pathology, we describe it in 5 different phases. Phase 1 being limited to the neocortex, phase 5 being very widespread, including involvement of the cerebellum. So second hallmark of Alzheimer's disease is tau, the neurofibrillary tangles. And so the neurofibrillary tangles, they start from the entorhinal cortex, spread into the, so here, this is the entorhinal cortex. This is Brock stage 1. When they spread into the hippocampus, it becomes Brock stage 2. If it goes into the adjacent temporal cortex, it's stage 3. If it goes into other areas of the cortex, it's 4. And then lastly, you take a section of the occipital lobe where you have the visual cortex and the adjacent called the peristriate cortex. So if it goes into the peristriate, then it's Brock stage 5. If it goes into the primary visual cortex, it's Brock stage 6. And neurofibrillary tangles, this is a picture of the hippocampus. So basically they are intracellular. So they will show up as these brown staining of taking shape of the neurons. So this is what a neurofibrillary tangle looks like. And based on what brain regions involved, you stage it from stage 1 to the most severe stage 6. Then another measure of plaques is to look at the neuritic plaques. So this constitutes the CRAT score. And there are different ways of doing this. So you could do it with Bushofsky's silver stain. So these look very similar to the amyloid plaques you see here, except that it is not antibody staining. These are silver deposits onto these structures. Again, you see a core and a rim of blank staining, and then another rim of hazy staining. So these are called neuritic plaques. You can also do thioflavin, which is kind of a chemical compound, and look at it under fluorescent microscope. Or you can just do tau, and then these label actually the the neurites that the axons that are damaged and surrounding the plaque. So the CRAD score is basically a quantitative score looking at whether you have no plaques, sparse, moderate, or frequent plaques. In addition to plaques and tangles, because of AD also has high comorbidity with Parkinson's disease or dementia Lewy body. It also has high comorbidity with a recently classified kind of pathology called limbic predominant age-related TDP-43 encephalopathy. So that's also different pathologies that we always evaluate when we get a dementia workup. So Lewy bodies are labeled by alpha-synuclein, and they are divided from the earliest stages is confined to the brainstem. Then they spread into the limbic system and finally into the neocortical. There is a special type of Lewy body disease called amygdala predominant, and this is usually comes with very severe Alzheimer's disease. So the contribution of Lewy body pathology in this case is unclear, but what you see is Lewy bodies confined to the amygdala. Then TDP-43 is normally a nuclear protein involved in regulating RNA splicing. So usually it will have a very light kind of hazy staining in the nuclei, but in pathologic conditions, it will form these very solid spherical inclusions in the cytoplasm. And it also spreads from the amygdala to the hippocampus and then to the neocortex. So we stage it accordingly. So these are the bread and butter, the most common dementia cases we get. Occasionally, we will get some more complicated and less common dementia cases. For instance, sometimes sometimes we get these cortical basal degeneration, which I showed the gross picture, that is characterized by profound asymmetry. These are characterized by tau and tau lesions, but instead of neurofibrillary tangles, they are characterized by tau accumulating in astrocytes. So here you are seeing that there is a astrocyte nuclei here, and then these are the distal processes of this astrocyte. This is called the astrocytic plaque, and this is characteristic for a cortical basal degeneration. Okay, another type of tauopathy is progressive supranuclear palsy. Again, this is the pathognomonic feature is tau accumulation in the astrocytes. Again, this is labeling, you see a nuclei of an astrocyte. And here it is labeling the proximal processes and a little bit of the cell body here. So these are called tufted astrocytes, and these are characteristic feature of progressive supranuclear palsy. Another lesion that is characteristic is called these coiled bodies. So these are kind of like comma-shaped type of staining, and these are like the short and stubby processes of an oligodendrocyte. So for CBD and PSP, they are characterized by tau accumulation, not in the neurons, but rather in the astrocytes and the oligodendrocytes. So this is what a neuropathology report would look like. Basically, we try to approach every case in a very standard way. We give a pathologic diagnosis, and it is then the job of the clinician and also the neuropathologist to communicate and try to come up with a integrated diagnosis and to see if the neuropathology and the clinical features match. But with regard to the neuropathology diagnosis, we just say it's Alzheimer's disease neuropathologic change, and then we give a score based on the staging scheme that I just described. Also, we give a measure of how much Lewy body disease there is, how much limbic predominant TDP-43 there is, and all other different types of pathologies that you can see in the brain. And then there are, then we look at the clinical presentation, and we can discuss with the clinicians whether this neuropathology makes and the clinical presentation matches, or whether there's any inconsistencies that we have to do more workup and more communication. So that is the neuropathology course, serving as a diagnostic service for the clinicians and family. But another very important function of the brain bank or the neuropathology core is as a biorepository. So at the time of autopsy, we take a sterile needle and go into the ventricles and to get the CSF. And then we also section the brains fresh. So this is one of the brains that John Irvin, who is the lab manager of the brain bank, he did. So these are very, very nice sections, and it really takes a lot of practice to do it. Here you can see like the caudate, the putamen, globus pallidus, external segment, internal segment. This is the thalamus. So you can see all of the internal structures really well. And he tries to do it in a way that matches up to the formalin-fixed tissue. Then he also sections the cerebellum and the brainstem. And all of this is, he brings it, he needs to dissect it as fast as possible, put it into 2-methylbutane, and freeze it down as fast as possible. We have 8 full -80 freezers plus 2 spare ones, and we house a collection of about 1,100 brains, another 400 CSF samples, and we also have DNA samples housed elsewhere. But really, the frozen brain tissue and the CSF is housed in the brain bank, and these can be used to support many kinds of different types of research. We have the vast majority of our brains are AD and other dementias. We have about 200 of other neurological disorders, including Parkinson's, various tauopathies, and then we also have about 200 normal controls. So in the last few slides, I just want to give everyone a type of a few examples of what kind of research we can support, and so maybe can help everyone think about how to utilize this resource. So because we have over the years collected over 1,000 samples, one of the most obvious type of research we can support is clinical and neuropathological studies. And this is a recent paper that I published looking at late limbic predominant TDP-43 encephalopathy and comparing it to other pathologies that are also caused by TDP-43, and then looking at the associations between late and other common pathologies. We are not just a standalone brain bank, but we are a network of many different brain banks across the world. So this is another study that we participated in, and this is another study on late. This is a collective data from 13 community-based or population-based autopsy cohorts. And so this is probably the largest study on late. So far, we have over 6,000 subjects, and we are looking at the association with various different measures, such as ApoE4, age, sex, Lewy body disease, different measures of AD pathology. We are a network of different brain banks, and we regularly contribute our data to these large consortium type studies. We are also recently going to contribute data on some genetic studies looking at a very rare kind of tauopathy called Pick's disease. So in terms of the biorepository, as I mentioned, we have CSF and frozen brain tissue. So we've been supporting studies to develop diagnostic assays. For instance, this is a study published by Andy West Group from using CSF and brain tissue samples from us, where they looked at the seeding activity of Lewy bodies and of alpha-synuclein, actually. Looking at the correlation between the seeding activity and the disease duration and progression of Lewy body disease. We also have other collaborations also using CSF as to develop diagnostic assays. So in terms of brain tissue, we've recently supported a lot of these single cell transcriptomic studies, or here I'm showing collaboration with Simon Gregory's group and Diane Cruz. We can also do these spatial transcriptomic studies where we take frozen tissue samples from different brain regions. Here we're selecting 4 regions spanning this entire spectrum of AD. For instance, entorhinal cortex is a region that's involved very early on, and striate cortex is one that's involved very late. And so we can look at the transcriptomic profile of these different brain regions that are affected at different stages of AD. And we can convert like our high level data into UMAP plots, and we can see that the gene expression profiles of these 4 different brain regions clearly segregate from the most severely involved entorhinal cortex to the least involved striate cortex. We can also go back, plot it on to the tissue sections to look at globally what are the differences between these different brain regions, but also zone in on different genes and see where they are expressed. For instance, here we're looking at the expression of GFAP, which is an astrocytic protein, and the different color codes describe the level of its expression. So these are just a couple of different studies that we can support because we have the tissue, we have the neuropathology data. We also have clinical and cognitive data on a subset of our participants. So that's also something that we could do to support your research. So lastly, I just want to point out that this is where we are. This is the Bryan Research Building on the Duke campus. We are on the 2nd floor. This is where everything happens, the brain dissections and all of our freezers and all of our samples are housed on the 2nd floor of the Bryan Research Building. And so with that, I would be happy to take any questions.
Hi, thank you so much for that talk. I'm Chelsea, I'm one of the research scholars. I actually had a question that was a little bit less about research opportunities, but more about like clinical applications. So I was really interested in the staining and things like that. Have there ever been a study that looked at like the different like tau and BRAC staging and like correlations in imaging and like clinical findings or things like that?
So there's been a lot of studies, both. So people have done studies looking at BRAC staging and correlation with the various clinical measures. They have also looked at correlation between BRAC staging and also pre-mortem imaging studies. So a lot of that has been done. I think what an area that may may warrant further development is to look at from a more of a clinical aspect is maybe to develop better tracers, like different PET tracers that are more specific to tau, and then different tau-based biomarkers for diagnosis. For instance, there's been a lot of discussion about using phospho-tau 181 to 17 in CSF for early diagnosis of AD. I think there's more more to be developed in that area. The other, in terms of more like the basic science aspect of it, is that the Brock staging is accurate in most cases. However, there are a small percentage of variant ADs. So they can have like hippocampal sparing variant. There's like limbic predominant variant, and they can have different clinical manifestations. There's also different types of tau antibodies. So we are using an antibody called AT8. So that recognizes specific epitopes on the tau protein. But there are also hundreds of post-translational modifications. And we haven't really looked into like whether they will label different types of tau lesions or not. So the short answer is yes, there's a lot of potential development in different areas, like improving diagnostics, or from the basic science perspective, understanding how the evolution of these different tau lesions.
Great, thanks so much.
Hi, Dr. Wang. I'm also one of the Caring Star Scholars. I'm one of the neurology residents. Thanks so much for your talk. I was really curious about the CRAD staging for the neuritic plaques. Mm-hmm. And I'm sorry if you'd mentioned this, but I'm curious to know what specifically that looks for or what you typically see for those who have a clinical diagnosis of Alzheimer's. Is the staging based on the severity, or is that based more on the location? I think you may have specified that localization is what helps with the staging.
Right. So there's three main measures of staging. So this is where I put down A3, B3, C2. So the three measures are tau phase. This looks at the distribution of plaques by amyloid beta immunostaining. Then there's the B score, which is the Brock stage. This looks at the distribution of neurofibrillary tangles by tau immunostaining. Then there is the neuritic plaque score. So this is called the CRAD score. And this is looking at the density. So it's not really distribution. So this is the density of these plaques. So the first two scores, A and B, look at the distribution. C looks at the density. Then there is like this complicated matrix where you convert like your A, B, and C score into low-level Alzheimer's disease neuropathologic change, intermediate level, or high level. And what we now know is that if you have intermediate to high level of neuropathologic change, that is usually sufficient to explain the person's dementia. So here's a few different scenarios. So if a patient has dementia and then there is intermediate level of AD neuropathologic change, then you can say that, well, this dementia is most likely caused by the AD. If the person has no or low AD neuropathologic change, then you have to go back and think about, what did I miss? Then you have to think about maybe this person has another type of dementia. If the person doesn't have dementia, but has a high level, intermediate to high level of neuropathologic change, it could be because we know that these neuropathologic change, they occur, they can take like decades to develop, and the onset of symptoms may lag onset of pathology by up to 10 years. So we might have just caught them at this window. So the short answer is yes, there are different measures that look at the distribution. There are also measures that look at the density of these different lesions.
Thank you.
Oh, I just want to add that there has been a lot of studies that look at these different staging schemes, and they found that the the cognitive phenotype correlates best with the Braak staging. And the second best measure is the CRAD score. The tau face doesn't really correlate that well, but still in routine clinical practice, we still use the tau face.
Hi, Dr. Wang. I'm Audrey. I'm also one of the Caring Star scholars. Interesting talk. I was especially interested in the section where you were talking more about biobanking and some of the promises in advancing our understanding of genomics, transcriptomics, and proteomics. I can imagine, though, that some of the preservation techniques in biobanking can affect the fidelity of those kinds of studies. And I was wondering if you could speak a little bit more about whether that's been a factor, or, if not, what you consider potentially some of the limitations within biobanking or future directions for research.
Right. So there's a lot of challenges, basically. So people have looked into this for many years. So one thing that could definitely affect the quality of your tissue is the postmortem interval, and so that's why the clinical coordinator is so important, because you want to have everything. You want to have a well-oiled machine to get. When you know of a participant's death, you want to get the body transported to the autopsy team and then get into the brain bank as soon as possible. So there's the postmortem interval. Then there's the freezing method. So here we use there's different methods of preservation. So in the past, what we did was we just sectioned the brain and then put them between like two metal plates that are cooled in -80, and then put them as soon as we can into a -80 shell. Now we've started submerging them into 2-methylbutane, and so we put 2-methylbutane is just like a solution that transmits cold very well. So you put a 2-methylbutane in a large styrofoam box filled with dry ice, and then you submerge in the tissue sections in that. That's another technique. And I think the one that's most labor intensive is like you use a liquid nitrogen tank, and then you have like this device where you put liquid nitrogen, and on top there's a hole, and then the liquid nitrogen vapor comes out, and you put your sections between two metal plates, put it into that chamber, and let the liquid nitrogen vapor cool it down. That's probably the fastest and the best measure, but it is very labor intensive. Also, you have to consider like how big the human brain is. So when you're looking at a section, the temperature between like the outside of the brain and the inside can be very different. The speed that you freeze it down, you can freeze the outside cortex almost instantaneously, but in the inside the structures, it can take like a few minutes. So that's all like different limitations. So people have been looking at, like, there's a kind of measurement called RNA integrity score. The higher the better. So you can have a very good RNA integrity score in the cortex, but very poor in the middle. And I think it's still something that people are trying to figure out. What is the best method of freezing? Of course, we know that if we can cut the tissue as thin as possible, then it's better. They can freeze faster. But then it also takes longer to cut the tissue. Then it's always a trade-off. So So, and also with all these like new omics technology, what it's currently still up to debate what measure is most important. So this is something that people are still doing doing studies on.
Hi, Dr. Wang. I had one more question. When you were talking about the the clinical coordinator and things like that, as like clinicians working in the hospital and things like that, is there something that we can do to try and connect like patients with clinical coordinators? Like, how does that kind of happen? And is there a way that we as clinicians can help make that happen?
Right. So now that we have a funded ADRC, so we have dedicated coordinators who are actively recruiting patients. If you have a patient that is interested in contributing to AD research, then I would say refer them to the ADRC. Occasionally, we get patients who just died of a neurodegenerative disease, and they were not able to make arrangements beforehand. There is a network called the Brain Donor Project, and it's run out of Florida by this person who used to be at Duke. I forgot the name, but if you Google Brain Donor Project, you'll find it, and it's an absolutely amazing network. So they have contacts. They have like a network of pathologists almost in every state, where if the family just calls this number, they can refer you to a facility nearby that is going to remove the brain and then ship it to Florida. Or there's a few brain banks that are part of this network. I think maybe 6, and then they're arranged by different regions. So I think in the Southeast, they all get shipped to Florida. And I think as a single institution at Duke, we don't have that kind of capacity yet, but luckily there is this brain donor network or brain donor project that's supported by NIH that's running out of Florida, and it seems to be functioning very well.
Great, thank you.
And I just want to add that we, as an ADR, so every ADRC has their own recruitment goals. And so oftentimes we get inquiries about like brain donations, and I always feel bad that for a large number of them, we have to turn them away. But it's just that every ADRC has has a different goal. So for instance, the Duke-UNC ADRC at this point, we're trying to recruit younger patients. So our target is people from 45 to 65, although we do accept up to 85 years old. But really what we're trying to do is to sign up people in their middle age and start collecting biospecimens and data during middle age and see what changes, what predisposes these people to develop AD. And we're also focusing on people who have a strong family history of dementia. So every ADRC has their different has their different study design and recruitment goals. So unfortunately, for a lot of people who just call in and are really eager to donate their brains, I usually just refer them to the Brain Donor Project.
That's really helpful to know. Thank you.
Any more questions? Okay. Well, thank you, Dr. Wang.
Okay, thank you for coming.